and welcome to this Genome Web webinar. I'm Ben Butkus, Editorial Director at Genome Web, and I'll be your moderator today. The title of today's webinar is Accelerating Drug Discovery in Cancer Research, Identifying Candidate Oncogenic microRNAs via Alternative Splicing, and is sponsored by Almaden Genomics. Our speaker today will be David Corey, Professor of Pharmacology and Biochemistry at the University of Texas Southwestern Medical Center. In addition, Mark Kunitomi, Chief Scientific Officer and founder of Almaden Genomics, will be joining us for the Q&A portion of the webinar. Attendees may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to David Corey of UT Southwestern Medical Center. Please go ahead, David.
All right. Well, good afternoon, good morning, or good evening, wherever you are in the world today. It's my pleasure to present this presentation on work that my lab has been doing with in collaboration with Almaden Genomics. Okay, so I'm a professor at UT Southwestern. I run a laboratory, and in the laboratory, we confront a couple constraints. One of them is the constraints of funding. You know, we have basically flatline funding, so we have to make sure that we use our resources as wisely as possible. We cannot miss opportunities. The other problem that we confront is it's becoming more and more difficult to get postdocs and graduate students to join academic laboratories. So we have to get the maximal productivity out of each person that we can. And one of the ways that we do that is to leverage the enormous expansion in our ability to get large data sets. And, you know, a couple ways we do that are by doing a lot of RNA sequencing and also mass spectrometry. So there's no doubt that those are wonderful new techniques. They give you vast amounts of data to analyze. You look at it just compared to 5 or 10 years ago, we're being handed unimagined amounts of data. But the problem is that we're really more comfortable with benchtop experiments. People in my lab in general don't know how to write code, or they don't know how to do it very well. They're not good at working with complex programs. And we're often beginners at the analysis of large data sets. So we have this problem here. The scientific opportunities are enormous. The opportunities for increasing our productivity so that we can stay on the leading edge of research are huge. But there's also an enormous danger of wasted effort, misleading interpretations, and irreproducibility, which I'm going to be talking about throughout this talk. So the question is, what are we going to do about it? Okay, so here's the basic problem. We're wet lab researchers. We're not computational biologists. What we do is we do experiments. We submit those materials for analysis. And in general, what happens is it basically goes into a black box that the wet lab experimenter doesn't understand. And what comes out at the other end is some kind of data analysis. And the problem is that the data analysis we do to form our hypotheses completely depends on what's going on in that black box. And let's keep in mind, what are the stakes going to be here? That analysis from when you submit the samples to when you finish might only take a few weeks, and someone in a core facility might do it for you. But then it might take a couple of years to actually test the hypothesis. And if that hypothesis is not really based on good analysis, you could end up wasting a couple of years, which means that you might not get that grant funded, your graduate student might not graduate in a timely fashion, your postdoc might not be able to go on with your career. So getting this right has really high stakes. Okay, so what are these challenges? Well, part of it is just the data is complex. The input is complex. Biology is messy. So therefore, it can be very, very difficult to work with. If you don't really, if it isn't transparent, it can be very hard to know what's signal and what's noise. There can also be a documentation gap. There might not be clear guides to understand how to analyze the data, or to go back and repeat the studies of others, or for others to go back and repeat your studies. Methods can be uncertain. You know, a lot of times when you go in the literature and you see how people have analyzed data, that experimental write-up is grossly deficient. And again, that makes it almost impossible sometimes to repeat the analysis. And finally, you can get different software versions, and that further complicates our ability to repeat studies. Okay, so how do biologists address these challenges? Well, I'd say that there's pretty much three ways to do it. The first is ignore them. And that's probably the most common way, is that you just do what you can. And all too often, people will get the results they want to get, and they will move ahead, they'll publish, and everyone will end up being confused. The next possibility is to collaborate with a computational biologist. And no doubt that can be wonderful. The problem there is twofold. The first is that the computational biologist does not usually understand the biological problem, and therefore just doesn't have that intuition about where the data is going to lead, where the diamond is going to be. They don't know how to see it. The other problem is that computational biologists are highly sought after. So your project may be their 10th priority. The final possibility is to do it yourself. But that requires having someone in your lab who knows how to code, who knows how to dig down and do the computational biology. That person is worth their weight in gold, but they are extremely rare. Okay, so let's dig into this a little bit deeper. So there's a skilled gap. It's no secret. Biologists are biologists because they like biology. They like doing experiments. If they were going to be coders, they would have done coding. So they often lack experience in coding and statistics. We do try to give our students a basic understanding of both of these things, but that's what they have, basic understanding. Okay, data handling, managing big, complex, and messy biological data, very difficult. And again, as I've said before, staying up to date with the latest tools can be very challenging. Okay, now let's return to this issue of collaboration. Fundamentally, this is one of the biggest challenges. If you have an experimentalist and a bioinformatician who are working closely together, that's the best case scenario. But how often is that really going to happen? In our experience, even when we do have these friendly collaborations, it's easy for little things to slip through the cracks. And those little things can then compound to missing thousands of potential pieces of data that could be significant. Okay, and finally, limited availability of high performance computing, and even knowing how to access it, if it is available. Okay, so it's important to think about what are the consequences. So part of this is informed by my kind of second job as an editor for nucleic acids research. And in that context, I've been seeing a few hundred submissions every year. And many of these submissions involve the analysis of large data sets. And unfortunately, what I see too often is too little appreciation on the part of experimenters that this data is actually difficult to analyze and that you have to justify that you're doing it right. So I like to call this do not try this at home, that these laboratories don't understand the limitations of the approach. And as a result, the data that they're pumping out and showing to the community, it may superficially look like something's interesting, but it's not really clear that there's anything there. And that leads to the second point, kind of a just trust me attitude that you find in many papers, that researchers don't know how to present data in a transparent fashion. And I think in a lot of cases, it is absolutely impossible for reviewers to have really reviewed it. It's just like, here, this is what we've done, trust us. They're not providing enough information for a reviewer to go in and really have any confidence. And for me, that's always a red flag that a paper probably should be rejected without review. And then finally, that leads to the key problem. I would say one of the big things facing science today is a lack of reproducibility. You know, reproducing a published experiment, let's face it, that's always going to be a thankless task. But it becomes impossible when protocols are garbled and the computational pipelines are not available or poorly defined. Okay, now it doesn't have to be this way. I got my PhD in chemistry, and chemistry for as far back as I've been around, if you try to publish a paper, there are specific guidelines that you have to meet before that paper will even be accepted for review. You have to meet certain criteria of molecular weight, elemental analysis, providing NMR spectroscopy, primary data of HPLC, all of these things, they have to be included so that they can be seen. Those are boxes that have to be checked. Probably what we need to do is move toward having computational data fill similar, rigorous, standard criteria so that if you read a paper, you can understand where the paper is coming from. And then conversely, if you're an industry and you're trying to find out whether the people working on a project have really squeezed all of the important data out of something and have not missed the critical piece of data that's going to form a hypothesis that's going to make you $1 billion, you're going to want to make sure that they have checked all of these boxes to make sure that the analysis was done properly. Okay, so how can best practices make reproducible genome analysis routine? Well, we need to develop transparent data analysis pipelines. To all the extent possible, these pipelines have to be understood by experimentalists. This should just be common sense, that if the people doing the experiments don't understand, even have a conception of how the data is analyzed, then they're not going to be able to get the most out of that data. They may form the wrong hypotheses, or just as bad, they may miss important hypotheses. And these pipelines have to be able to be used to reproduce results in other laboratories. Okay, so with that background, let's talk about the specific research project problem that we brought to our friends at Almaden for them to help us with the analysis. So this is going to concern RNA interference and unmet challenges in microRNA research. Okay, to give you the sort of big picture of backgrounds, there are two strategies for targeting messenger RNA and silencing gene expression. The first uses antisense oligonucleotides. This is the senior modality. An antisense oligonucleotide will bind to messenger RNA and inhibit its expression. And that's been commercially successful. There was a drug for ALS that was approved just a couple of months ago. The other approach that's at least as successful is double-stranded RNAs. With double-stranded RNAs, you introduce them into cells, they bind to the proteins of the RNAi silencing complex, and those proteins efficiently deliver the silencing RNA to a messenger RNA. This approach has proved very valuable. And as a result, the leading companies in this field are now valued at tens of billions of dollars. And striking clinical success is becoming routine for these molecules. There are now about 13, I think it's actually gone up to 14 now, approved nucleic acid drugs, with 2 or 3 new ones being approved every year, and we can expect that to increase post-COVID. Okay, but there's another way of thinking about drug development. MicroRNAs are small, naturally occurring RNAs that silence gene expression. In this mechanism, a protein called Argonaute protects the microRNA and facilitates recognition. In contrast to what we see with fully complementary RNAs, and that's the standard mechanism that a company like Alnylam would use, microRNAs have mismatches in the middle. They don't induce cleavage. So undoubtedly, powerful natural mechanism. One would think that if we could target these microRNAs, we could have a whole new class of drugs. But this is how many approved microRNA therapeutics have been developed in the 20 years that companies have been working to try to develop them. Nothing. So we have to ask ourselves, why has there been so much success with synthetic double-stranded RNAs, but there's been no success at interfering with naturally occurring microRNAs that function through exactly the same mechanism? And this is also even though we have an exquisite chemical toolbox for making compounds that will bind and block natural microRNAs. Okay, so why have they lagged behind? What do you need to know? The bottom line is that clinical success has demonstrated the power of RNAi in vivo. Just go visit Alnylam's webpage. Look and see what they have in the pipeline. No doubt that this is a powerful mechanism in mammalian cells. We also know that microRNAs have demonstrated roles in plants and animals. No doubt about that. And now let me give you one final thing to think about. There are thousands of papers citing microRNA that appear every year that claim to link a microRNA to an important gene involved in human health. So put these things together, and the logical conclusion would be that microRNAs are great drug targets, and we should be moving ahead with them. This just gives you a little idea of how many papers are published. There are literally thousands of papers every year that, if you plug in the word cancer and microRNA, will show up on a PubMed search. So it seems like this is a pretty mature field. Everything is pretty well understood. But that impression would be wrong. When you look at these many papers, most of the papers lack controls. There is little insight into mechanism, and there's an insufficient appreciation that microRNA function is complex. And in fact, if you go in and you try to reproduce most of this research, you're probably going to be very difficult for you. So we tried to do this, and in our hands, there was confusion in assigning miRNAs to function. We couldn't even reproduce the most high-profile experiments. So this suggests a fundamental lack of understanding of how these ubiquitous, naturally occurring regulatory molecules function. So this is what we were faced with. Explanation one. MicroRNAs possess a bewildering array of biological functions that are difficult to reproduce, or something is missing from our popular understanding of microRNAs. So I usually am a big fan of the simplest is always the best explanation. So I would favor explanation two. Okay, let's get in a little bit more of the complexity of microRNAs. Remember I told you there are thousands of studies per year? Those studies are usually based on the hypothesis that a single microRNA binding to a single messenger RNA is sufficient to control gene expression. But in fact, what's much, much more likely is that the way it works is that microRNAs bind in combinatorial fashion with different microRNAs binding close to one another. So I think you can now see how this recognition is getting ever more complicated, and that multiple microRNAs may contribute to repressing a target. Now, that doesn't mean that using these as drug targets is going to be impossible, but it means that people are going to need to take a much more thoughtful, quantitative, and mechanistic approach. Okay, this just gives you some data from our lab. This is a technique called ECLIP. And just to give you the quick overview, ECLIP is a technique where you use an antibody, in this case, an antibody against the critical RNAi protein Argonaute, to bind to an Argonaute that's associated with RNA within a cell. So it gives us a way to identify all of the candidate RNA in a cell that might be binding to a microRNA. And what you can just see on the left is that we get a couple big peaks at the c-MYC gene, where it looks like Argonaute is being bound. Each of those peaks is about as big as a microRNA, but all of those little dots is different microRNAs that have complementarity to those sites. So it gives you an idea that you have two microRNAs, but you got lots of microRNA candidates. So this is an example of how getting RNA sequencing data can give you the beginnings of where you want to start to get a hypothesis. All right, so we want to identify a therapeutic target. We need to know which microRNAs modulate a critical function. We need to know what genes are modulated and how does controlling them lead to therapy. And at the end of the day, we want to know, when does a microRNA play a critical role in health or disease? And the only way we can get that is efficient use of large data sets. So how do we use RNA-seq to understand human RNAi? And specifically, what I'm going to address today is not how RNAi can affect translation. That's kind of the canonical way, but something that's a little bit new. How might it be able to affect gene splicing? Okay, so we had been using large data sets to try to find natural microRNA-mediated control in HCT 116 cells. And this ended up being a little bit frustrating. No matter how careful we looked, we were not able to find any clear examples of translational inhibition. But we were able to find a few examples of regulation of splicing when we looked at RNA that was harvested from the nucleus. So we asked ourselves, we're not saying in normally grown cells that microRNAs are doing very much. Is it possible that microRNAs are more important when cells are pushed to extremes, when they need to have some kind of environmental stimuli to make the cells respond so that they actually need the microRNAs? So we asked ourselves, what happens when HCT 116 cells are grown to overconfluence, a very simple environmental stimuli? So this was a project that was taken on by Crystal Johnson in the laboratory, who'll actually be getting her PhD at the end of this week. She examined cells that were grown to high confluency in normal 2D cell culture, in three-dimensional organoid culture, and in primary tumors. So in 2D normal culture, HCT 116 cells will just grow and stack upon themselves. That's very clear if you look at them under the microscope, and you can quantitate that using a cell counter. Very easy, very reproducible method. And when Crystal did this, she went from, at normal confluence, Argonaute 2 mainly being in the cytoplasm, to, at high confluence, it's almost entirely in the nucleus. So you get a many-fold shift to the nucleus when you grow cells to a high density. A simple environmental change dramatically changes RNAi in these cells and in other cells that we tested. So what are the consequences of overproliferation on microRNAs? What happens to the microRNAs and what happens to microRNA prevalence? Okay, so for this, we needed to prioritize which microRNAs we were going to look for. Okay, not all microRNAs are created equal. One of the things that's often missed in microRNA research is that microRNAs that are more highly expressed are likely to be the ones that are the most active inside cells. So we used RNA-seq to identify the most abundant microRNA families, and we used our eClip to identify genes where Argonaut was likely to be binding to. And then we combined these two things and asked, what are the sequences that the most highly expressed microRNAs were likely to associate with? Those sequences are going to be the best candidates for microRNA action. So one of the things that Crystal immediately saw was that when you shift microRNAs to the nucleus, and she's showing this on the left, many of the genes that we would expect to be candidates for normal translational repression become upregulated. So this leads to the simple hypothesis that moving Argonaut protein to the nucleus just removes a translational regulation in the cytoplasm. So that's one thing that may be occurring. Okay, now let's go on and see whether or not we observe these same things in more complicated systems. So we collaborated with Rolf Brecken's lab on my campus to look at three-dimensional cell culture. And here it was even more striking. When we harvested the organoid cells, we found that Argonaut protein had entirely shifted into the nucleus. Nothing was observed in the cytoplasm. So this then encouraged us to go and look in tumor samples. So Crystal looked at 29 different tumor samples. And as you might expect, if you've ever looked at tumor samples, the effects varied. But in all of these samples, when we looked at the adjacent benign tissue, Argonaut was primarily in the cytoplasm. In fact, overwhelmingly in the cytoplasm. However, when we looked in malignant colon tissue, we found that Argonaut was associated with the nucleus and even more with chromatin. So that was in about 8 of the 29 tissues we looked at. So not all of them, but in a substantial percentage of colon cancer tissue, what we were observing was this dramatic shift of the RNAi apparatus from the cytoplasm into the nucleus. So we don't right now know what the consequences of that are, but it's something we're going to need large data sets to figure out. All right, so I've just told you that we're seeing Argonaut in the nucleus now. What might it be doing in the nucleus? One explanation is that you're just sequestering it in the nucleus and it's not doing what it normally does in the cytoplasm. That's possible, and we have evidence for that. But let's just think about what we have here. We have Argonaut bound to a small RNA. I like to think about that as ribonucleoprotein complex. You have an RNA domain, the microRNA, that directs specificity. The protein domain protects the RNA, promotes its binding to the target, and mediates protein-protein interaction. And all of this is programmable by the microRNA sequence. So in theory, you can recognize almost any sequence inside a cell and control the expression of almost any gene. So what might happen if this programmable RNA recognition machine might bind near a splice site. So let me remind you, the dogma is that Argonaute microRNA represses gene expression. Knocking down Argonaute would be expected to de-repress gene expression. Okay, but the question is, might it also affect splicing? So what do we need for this analysis to really make it work? And remember, we're going against dogma now. Our results have to be transparent. They have to be clear. Also remember that we're going to need to chase down any hypothesis with months or years of experimental research. So that, as I mentioned at the start, the stakes are really high. We need an efficient workflow to analyze the effect of increasing cell density. We need to understand how increasing cell density affects RNA splicing. And it's critical, I can't stress this enough, that we have ways to prioritize genes, identify the best handful of genes before we start time-consuming bench science. Okay, so this is just to give you some of the experimental justification for thinking about why we believe splicing can work. This is just a splicing model system in a transgenic luciferase expressing cell line. And what it shows is that when we target double-stranded RNAs to a splice site, we're able to efficiently change splicing in this model gene. We've also been able to use double-stranded RNAs to do the same kind of splicing switches antisense oligonucleotides can use for changing the splicing for muscular dystrophy or spinal muscular atrophy. So this seems to be a general approach that we can achieve using synthetic RNAs. Very robust, and it seems very reasonable to me to believe that natural microRNAs would use this mechanism to try to change gene splicing. But the question is, do our large data sets show that this is really happening? Okay, so basically, to summarize what I just said, what are the potential consequences of risk nuclear localization rather than have Argonaute in the cytoplasm, have it in the nucleus? We could regulate transcription, we could regulate alternative splicing, we could possibly regulate DNA damage, we could possibly regulate chromatin organization. There are all of these fantastic hypotheses that we can pursue now that we know that the RNA apparatus is a nuclei. Okay, so here's where we start with Almaden, and let me just briefly tell you what our experience has been. So, what does my lab need? Okay, well, I mean, ideally, we would need a full-time computational biologist working next to my experimentalists, or they could be rolled up in the same person. We don't have that at the moment. So what we really need is to be able to do some of this in our own laboratory so we can wrap our heads around it and apply our own intuition to the analysis of large data sets. That would really help to not need any coding, to have a drag and drop approach, which is highly intuitive, definitely for it to be transparent so that the people in my lab know what is going on. It should be intuitive, and it really needs to enable independent use. You can't have someone from my lab going for help every couple days when they need to do something. Okay, so this is kind of the big picture of what we get from Almaden. In this case, we explained to them what we needed, and they provided a custom computational pipeline work-through flow that would allow us to work. And this is just the overview. How I can see this developing eventually is them packaging a number of these fully developed pipelines for different commonly used bioinformatic needs directly to users for them to pick and choose in what they want. In any case, this is what they presented to us. Very easy to use, very intuitive. Let me blow it up on the next slide. So what you see here is that we just have our databases or our data collections. We plug them in on the left, and on the right, we get outputs in the variety of ways in which we might want to display our data. So all Crystal or anyone else in my lab needs to do is change which of these data sets are being compared to one another, and the data will come out. Very simple interface. Okay, now, but that doesn't mean that the coding is entirely hidden. At the top level, that will occur on a Jupyter notebook, which we do have some access to. And as I said, the people in my lab do have at least the beginning of some coding experience. And the nice thing about the Jupyter notebook is that they can go in it, they can access some of the code, and they can access explanations of what the code is and how it's used. So for the people in my lab who have some ability to analyze these things, it allows them to peek under the hood, to demystify things, to make the analysis even more transparent than it already is. Okay, so here's our experimental setup. We grow the cells to a high density. We spin them down. We separate them with cytoplasm and nucleus if we want. We harvest the RNA, and we go to sequencing. We obtain the data sets. And then, rather than have to wait 3, 4, 5, 6 weeks for our core to analyze it, we go directly to genome and analyze it ourselves. Okay, so we're able to look at different types of alternative splicing results. We're able to look at them by the number of events. So, that's very useful to us because we don't want to mix up the different kinds of alternative splicing results. We want to look at them individually, so we're not comparing apples and oranges. Again, it's all in this process of trying to winnow down the candidates so that we have intelligent reasons to focus on only the best ones. Okay, so when we do this, for example, what you might be interested in is only doing results on day 5 and day 7. And here you find that we go from a couple thousand to only 700. Okay, which candidates have Argonaut-2 bound targets with microRNA seed matches? We look at the computation for that. And now we're winnowed down to even more. It's only down to 192 now. So now we're getting into the range where we can start to look at these individually, look at the data, and see which ones are going to be best. Okay, so now what we might want to look at, well, how close are these bound complexes? Are they to a particular splicing event? Well, let's maybe form a hypothesis that they're going to be close to a splicing event. As you can see, we can see those, and we can identify which ones those are. Okay, and one of these splicing events is a gene called RAD51C. It's one of the most dramatic ones, so that's why I'd like to spend just a minute talking to you about it today. So RAD51C has an interesting sort of history. It's involved in DNA damage. So you might think that it could be involved in cancer responses to overproliferation. And this is a sashimi plot commonly used to evaluate splicing. Now, some of you may not have seen sashimi plots before, but basically the top 3 pieces of data are, look at what's happening at day 3, when you only have a normal confluence of cells. And what you see, there's a large amount of splicing change here. But then when you get out to day 5, it's dramatically changed. That's all gone away. And RAD51 is being spliced in an entirely different manner. So we're only beginning to study what the consequences of this change in RAD51 splicing are. But this is exactly what we as experimentalists are looking for: a clear change, easily detected, so that we can move very quickly from getting our RNA sequencing data to having a hypothesis that we feel confident enough that we can expend resources in the lab to test. Okay, and finally, just to point out some of the functions of RAD51: protein DNA complex assembly and cellular response to DNA damage stimulus. So seems like a pretty good candidate that our computational data and our work with genome has obtained for us. So obviously, I don't do any of this myself. Most of this has been done by my talented graduate student, soon to be PhD, Crystal Johnson. So, I asked her for some thoughts about this that I could share with you. So here's a couple quotes. It is starting to feel almost impossible to exist as a biologist in 2023 without some ability to analyze your own large data sets. And Genome allows me to analyze my own RNA sequencing data in less than a day. And I will tell you, for the last 4 or 5 years, I've been telling every young person who comes into my office that in their career, it is essential that they learn how to analyze these data sets. They will not be able to succeed as scientists without that skill. The other quote was that she's never taken coding classes, yet she can use the builder to quickly plug my data into splicing workflow and get publishable figures in minutes. So Genome allowed her to cut out the middleman to get results immediately. And, that's hugely helpful. Now, don't get me wrong, we're always going to need in-house computational biologists. But to the largest extent possible that we can do things without them will allow us to efficiently move forward with our science. So finally, I'd just like to thank the people in my lab, and I'd especially like to thank Mark and the other folks at Almaden Genomics for giving us the privilege of beta testing their platform and working with them to make it more efficient, and hopefully soon for them to transform this into something that can become very commonly used, where my university will be able to buy a license with a suite of different pipelines that my colleagues and I will be able to use routinely. Finally, I really appreciate your attention. I understand these webinars are a drag, and I'm very thankful for you spending a little bit of time with me today. Thank you, everyone.
Thank you, David, for that excellent presentation. We will now begin the Q&A portion of the webinar, where we'll be joined by Mark Kunitomi, CSO of Almaden Genomics. A reminder to attendees that you can type a question in at any time through the Q&A panel, which appears on the right side of the presentation. Please give us one moment while we gather questions. Okay, our first question. Have you ever tried to sample these cells in vivo to see if you will observe the same phenotype?
Right. Well, that's something that we want. That's something we want to do. And again, we're hoping that our experience with the Almaden pipeline will make that easier. The next obvious step would be to do the sequencing on the tumor cells. And I bet the person asking this question is very knowledgeable. They'll know that there's a lot of heterogeneity in tumors, and the data is going to be noisier than we get in mammalian cell culture. So that exact experiment is going to be our next step, and it's going to be the next challenge for Genome.
We might have to go into some single-cell RNA-seq, David.
Well, ultimately, that's where, we would want to go, because there's going to be heterogeneity at the single cell level. But, first, I would just say, let's see what's going on at the level of bulk cells before we take that additional step. Fortunately, we're at UT Southwestern. We have excellent access to patient tissue, here. So really, that's not the limitation. The limitation is our ability to analyze the samples. Great question. Thank you.
Thank you. Our next question: do you ever use long-read sequencing to assess splicing? And as a follow-up to that, do you feel like there are sufficient bioinformatic tools available for that?
I certainly can't comment on whether there's sufficient bioinformatic tools available for that. I think we're agnostic about what approaches that we take. Ideally, we would use different sequencing approaches. Look at it from my perspective as an experimentalist. We already have our experimental systems. We already have our cell lines. We have our battery of knockout cell lines. We have our access to organoid culture and tumor tissue. We have all of that. It makes sense to use all of these sequencing approaches and see what we get, so that we get the most out of our kind of fundamental experimental tools. But again, that all gets to the sequencing bottleneck of being able to analyze the data intelligently and form hypotheses about it. So Mark, I don't know if you want to address whether long-read sequencing would pose any difficulties for you.
No, I think that using long-read technology could be a really good application here in terms of being able to differentiate, especially if it's kind of complex splicing. And that would definitely be a challenge we'd love to bring on to the platform. So, for you and for others who want to be able to do long-read analysis, it's someone we'd love to chat with.
Thank you. Let's see, one moment while we gather questions, please. Okay, our next question. What are the options? I apologize, this question is for Mark specifically. What are the options for open source coding? Could a grad student, for instance, use their own code in your environment?
Yeah, absolutely. So one of the fantastic things about the Genome platform is that it's extendable. It's very easy as long as you have a containerized code to bring it in to the Genome platform. And there's actually a feature that allows you to do that in kind of a graphical way. And you can use that to bring in your own open source code. You can use it to bring in any open source code as long as it's containerized with a Docker image.
Thank you very much. Our next question. Do you think that nuclear localization of Argonaut, even though it's real and functional, is a property of overconfluent or stage 4 metastatic cells that have such diverse genomes and morphologies that they don't look like a human cell anymore?
That's a great question. And obviously, I don't know the answer to it. I think for nuclear Argonaut, what our work suggests is that people who have cells that are under stress, undergoing differentiation, diseased, developing, don't take it for granted that Argonaut is in the cytoplasm. Under these conditions, it may be doing things that you don't expect and that you have to find out for yourself. So I guess basically the hypothesis is that in many different systems, there may be untapped science. We were
Yeah, I
surprised.
mean, I think,
Let me put it that way.
well, and I think that's why it's so important that, your research highlights this in three different systems, right? It's not just cell culture, going from monolayer to overconfluent, because I think that in and of itself, that was the sole piece of evidence. You'd have to be look at that with a lot of scrutiny. But when you see it in tumor tissues and others, where it's very clear that AGO2 is transited to the nucleus, that really does pique the interest of, well, what is it doing there? As you said, there's still more to be done there, but this seems to be a reoccurrent phenomenon.
Right. And again, it's why it's frustrating when we get hung up spending time on analyzing data and not doing it efficiently. We can't move on to questions like that. It's frustrating. So that's why we need efficient and transparent pipelines, so we can get our answers in this system and then move on to other systems and find out how really general this is.
Thank you. We do have another question that is sort of along those same lines. Can Argo shuttling to the nucleus regulate nuclear non-coding RNA functions such as chromatin interactions?
Yeah, a fantastic question. That's another project in my lab right now, and it will involve sequencing RNAs that are associated with chromatin to try to prioritize candidate RNAs where that regulation might occur.
Yeah, I think there's a lot of great evidence that you have with the ECLIP data that kind of says, AGO2 is definitely sitting on multiple places within the genome, and how that's affecting chromatin state, I think, is another really intriguing question.
Thank you. So next question is seems to be a little more of an overarching question. What are the larger implications of this? Is every component of the risk complex currently known to be nuclear, or aside from AGO2, is the jury still out on that?
It's complicated. Crystal has been looking at that too. To take just one, so first of all, Crystal's work is all up on bioRxiv. It was well reviewed at Nucleic Acid Research. It's in revision, so you can trust what she has on bioRxiv. Just go and look at Corey and Johnson. TNRC6 is a scaffolding protein that's a critical organizer of RNAi function. It binds to multiple argonauts and assists in their cooperative action. When cells grow to high density, TNRC6 expression tends to decrease. So that effectively changes what its concentration is going to be in the nucleus and the cytoplasm. So yeah, lots of things can change. And it's a complex dance of function that we're only beginning to understand.
Thank you. That concludes the Q&A portion of the webinar. If we didn't, I apologize. We do have another question rolling in if we have a few more moments.
Sure.
Our next question is the localization of other splicing binding proteins. Let's see if I can, I think this is the local.
What the question
is. Yeah,
is.
you can see this.
Yeah. That's a great question. I can just say we haven't looked at that yet. It's reasonable to think that those might well change because there's been such a dramatic change in argonaut localization. And also by extension, not just splicing proteins, but other nuclear proteins may change depending on how they associate with argonaut in the nucleus. Yeah, definitely something that we need to look more at.
Thank you. And then we have another question from Mark coming in. Can you speak a little bit to some of the other pipelines available in genome that were perhaps not touched on in this talk?
Yeah, so we have a whole suite of RNA sequencing, RNA-seq analysis workflows to do, of course, alternative splicing, but also, your standard differential expression, looking at gene set enrichment analysis, looking at pathway analysis. We also are coming out with a whole suite of single cell RNA-seq and other omics workflows. They're going to be released over the next couple of months. And then, of course, you can do some of the other basic functionality in variant calling, variant imputation, other forms of genomic analysis. But we're always happy to receive inquiries and kind of go through what the capabilities of Genome are today, as well as look to develop workflows that meet your custom needs. See if there's a pilot that we could kind of do there.
Great, thank you, Mark. And that does, in fact, conclude the Q&A portion of the webinar. We would like to thank our speaker, David Corey of the University of Texas Southwestern Medical Center, as well as our sponsor, Almaden Genomics. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of the webinar or you would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.